Masurer got his PhD from University of Rouen in 2006 in pharmaceutical chemistry, and the title of his thesis was Synthesis of Functionalized Cyclodextrin for the Catalytic Purification of Organophosphate. His talk today is entitled Development of Kallikrein-related peptidase inhibitors, and actually this is a very important topic, particularly for the treatment of multiple sclerosis. So Professor Masurer, you are most welcome. Thank you for joining us today, and the floor is yours.
Okay, thank you for this kind introduction, and thank you very much for this invitation to be, and I will be very pleased to be here tonight with you. Did you see my presentation? It's okay, or you need that I put it on full screen? It's okay like that or not?
No, we cannot see it now.
Ah, you cannot see it. Okay. And here. No, not.
No, not yet.
Not yet. Okay, I will try another thing. Okay.
Yeah, it appears now.
Okay.
Now, yeah, now.
It's in full screen or not?
No, no, you need the full screen. Please press this F9 or this, yes, this icon, please.
It's okay, like that?
Yes, very nice.
Okay, sorry. I will just find a stilet.
Yes.
Okay, sorry. Let's start. I will present you today our results concerning the development of Kallikrein inhibitor. So before showing you this result, I will present you some general information concerning the Kallikrein family, and then I will present you our result concerning the development of inhibitor of Kallikrein 7 and finally against Kallikrein 6. So Kallikrein constitute a group of enzymes and more precisely a group of serine proteases involved in the regulatory mechanism of many biological processes. So the first characterized Kallikrein is plasma Kallikrein, also named KLK1B, and this Kallikrein is known to be involved in the formation of kinin, a group of peptides that are cleaved from kininogens. So the most known member of this kinin family is probably bradykinin, which may cause a lowering of the blood pressure, but also is involved in inflammation and in the mechanism of pain. So beside this plasmatic Kallikrein, there is also the tissue Kallikrein, named KLK1. This enzyme is also involved in the kinin formation, but this time in tissue. So related to the structure of this Kallikrein 1, 14 other Kallikrein named Kallikrein-related peptidases were then discovered and are called KLK2, 3, etc., until KLK15. So all genes that encode the Kallikreins are localized in the same arm of chromosome 19, and these genes share common characteristics. So all genes encode a single polypeptide chain as a pre-pro enzyme. And this pre-pro enzyme was first activated by removal of the pre-peptide, leading to the pro enzyme, which is excreted from the cell. And then this pro-enzyme could be activated into the mature form. This activation is done by various proteases or metalloproteases, or serine proteases, sorry. So once activated, kallikrein employs the classical catalytic mechanism of serine proteases to hydrolyze some peptidic substrate. So the peptidic substrate of kallikreins bind to the active site of the enzyme in such a way that the carbonyl group of the cis bond is positioned near the nucleophilic serine. And this serine could attack this carbonyl group, leading to the tetrahedral intermediate, which could then evaluate into the acyl form of the enzyme by removal of the first part of the peptide. And then the water molecule could hydrolyze the ester bond, leading to the recovery of the catalytic serine, which could be able to hydrolyze another molecule of substrate. This kallikrein proteases exhibit trypsin-like or chymotrypsin-like activity. So the large majority of kallikrein are analog of trypsin. So they can cleave a peptide bond after a positive charge amino acid like lysine, arginine. And kallikrein 3 or 7 are chymotrypsin-like proteases and cleaved the peptide after an hydrophobic residue like phenylalanine or leucine, for example. So in all tissue kallikreins, the substrate specificity depends also on the amino acids located more distant than the cleavage site. So this enzyme recognize their respective substrate via extending substrate enzyme interaction. And each residue of the substrate binds to a corresponding subpocket, as you can see here. And as proposed by Schechter and Berger, each amino acid of the substrate are named P4, P3, etc. And the cleavage site is located between the residue P1 and P1 prime. This residue occupied respectively the S1 and S1 prime subsite. So in addition to their substrate specificity, so these enzymes are not equally distributed in the body. So some kallikrein are exclusively expressed in a single tissue, like, for example, kallikrein 3, which is also known as a prostatic specific antigen. And this enzyme is secreted only in the prostate. Some other kallikrein are expressed in a restricted number of tissue. For example, kallikrein 5 is expressed in testes, breast, and skin, whereas others are expressed in many tissue. So this kallikrein can function alone or most often participate in proteolytic cascades to mediate crucial physiological processes. So in normal condition, various physiological mechanisms are involved in the fine regulation of the expression and of the activity of kallikreins. But in case of deregulation or overexpression, some pathologies could appear, and this specific kallikrein could constitute attractive targets for medicinal chemistry. So this is what I want to show you today. Sorry. So to begin, I will show you our results concerning the development of inhibitor of Kallikrein 7. So this enzyme is largely expressed in the skin and is implicated in the proteolytic cascade involving several Kallikrein, as you can see here, Kallikrein 5, Kallikrein 8, also Kallikrein 14. And this proteolytic cascade led to the degradation of some protein, DSG1 and DSC1. So these proteins are adhesion proteins that connect corneocytes together. So this proteolytic cascade is involved in the desquamation process, so it's totally normal. And this proteolytic network is controlled by a physiological inhibitor named LECT. So in case of deregulation of this network, overexpression or mutation in genes coding for the inhibitor, LECT, we could observe some skin disorders like dermatitis, atopic dermatitis or psoriasis, and in more serious cases, overexpression of Kallikrein 7 and total removal of this inhibitor led to this syndrome, the Nisserson syndrome, which is a lethal pathology that combines ichthyosis, atopy, and several other problematic issues. And also, this Kallikrein 7 is also involved in metastatic dissemination because overexpression of this Kallikrein 7 could reduce cell-cell adhesions and increase cell dissemination, especially in ovarian, prostatic, and pancreatic cancers. So several synthetic inhibitors of this enzyme are known and have been reported in the literature. We can cite this peptide derivative incorporating a chloromethyl ketone group, and a co-crystallized structure have been obtained with this inhibitor. But this inhibitor is not a selective one for Kallikrein 7. It could inhibit other isoforms, and it led to an irreversible inhibition, as you can see here, by alkylation of the histidine group of the catalytic triad. There is also heterocyclic derivatives which have been reported to be able to inhibit this enzyme. For example, coumarin 3-carboxylate derivative, which are also irreversible inhibitors, or isomanide derivatives, which are reversible inhibitors. But this structure incorporates some amino acid residue, which could lead to some problematic issue concerning their stability against proteases. So our aim is to identify new heterocyclic derivative without incorporating some peptidic or amino acid residue. So we started our program by screening an internal library composed of 65 different compounds which were designed for other programs. And among these 65 compounds, we obtained first hits of this family. So this family is pyridoimidazodiazepinone. And we observed a moderate inhibition with an IC50 around 50 micromolar. we show that this inhibitor is a competitive inhibitor, as you can see here in the Lineweaver-Burke plots using various concentration of the inhibitor and also various concentration of the substrate. We also determine that this inhibitor is a reversible one. So we started our program by realizing some structure activity relationship to identify a crucial position for this compound. So we plan to synthesize three different series. The first one is an urea series in order to study the influence of the aromatic group here by remove it. In the second series, we want to study the influence of position 2, 3, 4, and 5 of the diazepine ring. And finally, in the third series, we open the diazepine ring to evaluate the influence of this heterocyclic core. All series could be synthesized from a common intermediate here, a diamine derivative, which could be obtained by acylation of the 2-amino imidazopyridine. So let's start by the synthesis of the key intermediate. So in a first step, we protected the 2-amino pyridine by a tosyl group using tosyl chloride in pyridine. After we alkylated the nitrogen position of the pyridine, which offers this compound, which could be then cyclized using anhydride trifluoroacetic anhydride, to synthesize the imidazopyridine compound, where the 2-amino function is protected with a trifluoroacetamide group. So then this trifluoroacetamide group is hydrolyzed using sodium hydroxide to offer the free amino compound. This derivative was then acylated using Bock amino acid and coupling reactant. In this condition, we didn't see any N-acylation, but only C-acylation, as you can see here. This result could be explained by the N-amine reactivity. So the doublet of the nitrogen could increase the electronic density of this carbon, which could then attack the activated ester, leading to this C3 acylated compound. Then the Bock group could be easily protected using acidic medium. And could be this intermediate could be used for further modification. So in the first series, we remove the aromatic group to introduce an urea function. So to synthesize this compound, we react this diamine compound with carbonyl diimidazole at room temperature. In this condition, we obtain the targeted diazepine dione compound, but in a mixture with another compound, which is activated intermediate. So to prove the position of acylation, we introduce at the end of the reaction benzylamine, and we obtain the corresponding urea. And we prove the position of the acylation by NMR. We obtained an HMBC correlation between this hydrogen and this carbonyl group. Finally, we increase the temperature of the reaction, and in this condition, we obtain only the targeted diazepinone. We also synthesize various compounds by modifying the nature of the substituent in position 4 by introducing various amino acid lateral chain, as you can see here. Concerning the series 3, the open one, it could be easily obtained by amidification of the amine using carboxylic acid and coupling reactant. We introduced various aromatic group. We also tested heterocyclic group, and we change also the distance between the aromatic group and the carbonyl of the amide. Finally, concerning the diazepinone series, we studied position 2, 3, 4, and 5, and we begin by the position 2. So these compounds could be obtained by cyclization of our key intermediate using corresponding aldehyde, and the GEM diamino compound could be then oxidized using iodine and lead tetraacetate to offer the diazepinone here. We introduce various alkyl or aryl substituents, and we focus our study on the introduction of various methoxy group or dimethoxy or 3-methoxy phenyl groups. Concerning position 4, we also modify this position by introducing various amino acid lateral chain, and we synthesize also the enantiomer of our first hit. And finally, in this series, we introduce a methyl group in position 3 using Boc methyl phenylalanine. And then we reduce the carbonyl group of the ketone using sodium borohydride to offer the corresponding alcohol as a mixture of diastereoisomers. So then we evaluate all compounds at 50 micromolar, and most active compounds were further characterized. So enantiomer of our hit compound AE is inactive. We observe a loss of activity. So for all other compounds, we maintain the S chirality, stereochemistry. We also observed that when we removed the toluene group present in our first hit and replaced it by a carbonyl group, we also observed a loss of activity. We try several substituents, and we only find some activity with the dimethoxy series, and we could increase the potency in the case of 3,4-dimethoxy phenyl group. When we change the dimethoxy by the corresponding hydroxy group, we observe the loss of activity. We also show that the alkylation of position 3 is not favorable, as well as the reduction of the ketone, as you can see here. And finally, all open derivatives are less or not active. So to resume this structure activity relationship, only 3,4-dimethoxy phenyl group could increase the activity of our hit. This compound was characterized as a competitive inhibitor, as you can see in the Lin-Weberberg double reciprocal plot. And this compound is also a reversible inhibitor because dilution of the inhibitor enzyme complex by a factor of 40 led to the recovery of the enzymatic activity. This compound was also tested on 3 other Kallikrein to test its selectivity. and at 50 micromolar. And in this condition, we didn't observe any significant inhibition. And we determine his constant of inhibition to be 27 micromolar. So to conclude for this this part, we are not able to increase more the potency of this compound. So we didn't go further in the characterization and the activity of our compound. And for the moment, we stopped this work and we moved to another isoform, Kallikrein 6. So this enzyme is particularly abundant in the central and peripheral nervous system and is mostly secreted by oligodendrocyte, macrophage, and astrocytes. Kallikrein 6 is a trypsin-like protease, and it is involved in the degradation of myelin-basic proteins, and therefore is involved in the process of demyelination and remyelination. So this enzyme plays a crucial role in multiple sclerosis. This enzyme is also implicated in neuroinflammation after activation of protease-activated receptors. And it has been demonstrated that Kallikrein 6 is upregulated in active lesion of patients suffering of multiple sclerosis. Its level is elevated in the serum of patients suffering of multiple sclerosis. As I say just before, Kallikrein 6 mediates inflammatory response in the CNS through activation of PAR receptors. And also it has been demonstrated that Kallikrein 6 promotes a significant decrease in neuron number, as you can see in these two pictures, control and after addition of Kallikrein 6. So, this enzyme is also involved in a proteolytic network, and other proteases are also involved in the development of multiple sclerosis. Kallikrein 6 could be activated by the plasmin, and Kallikrein 6 could activate Kallikrein 1. Both proteases could lead to neuroinflammation, axonal degeneration, and also BBB disruption. Altogether, these proteases are involved in the development and in the evolution of multiple sclerosis. So the development of compounds which could inhibit Kallikrein 6, as well as other proteases involved in these proteolytic networks, could be interesting as a new strategy to treat multiple sclerosis. So in the literature, some inhibitors of Kallikrein 6 have been described. So peptidic compound, but also covalent inhibitors, as well as small molecules. This is small molecules which retain our attention, and in particular this compound, because it's shown a micromolar potency, and only few derivatives of this series have been studied, and a co-crystallization of this compound with the protein is available. So the co-crystallization revealed that the inhibitor occupies the active site of the enzyme, and the benzylamine group here occupies the S1 pocket. So the primary amine, we can see it here. I have normally a small motion, but it didn't work here, but no problem. So the primary amine interact with a carboxylic group, and we can see here that the phenol, the hydroxy group of the phenol interact with some amino acid as well as the NH of this compound. So, okay, sorry. So you can see here the picture, the motion. The benzylamine is in the S1. No, it didn't work. There is a problem. Okay. Sorry. Can you? I have a problem with my presentation. Can you still hear me?
Hear you,
Yes,
yeah.
yes,
Yes, we hear you, yeah.
yeah.
Sorry, I have a problem with.
Skip and come back. Just skip and come back.
Yes. Okay, I will. I will open it. Sorry for this problem. Hop. Okay. I hope. Okay. So concerning this inhibitor, we observe that the free amine is implicated in ionic interaction with the aspartic acid in the bottom of the pocket. And we also observe other interaction with the hydroxy group of the phenol and also between the NH group and all the key amino acid of the enzyme. So we decided to keep constant this part in red in this figure, and we want to synthesize various compounds by modification of all position of the phenol part, but also by introducing some naphthol residue. So concerning the synthesis of phenol derivatives, we started from this benzylamine compound, which is protected using Bock anhydride in basic medium. Then the nitro group is reduced using hydrogen to obtain the corresponding amine. And then this aniline group is then coupled with salicylic acid using a coupling reactant to offer the amide derivative, which could be deprotected using hydrochloric acid. So we prepare a set of compounds by varying the position and the nature of the group, the substituent. So we observe that when there is no substituent, the inhibition at 10 micromolar is low. We observed an increase of inhibition when we introduce an alkyl group at position 5 or at position 4, as you can see here and here. So these three compounds were then further characterized, and we determined the IC50. The most active compound, MIT8, revealed to have an IC50 around 2 micromolar. So different concentration of the inhibitor and were tested using different concentration of the substrate, and the Dixon representation was drawn. And we can observe a variation of the Michaelis constant here, indicating that the MIT8 is a competitive inhibitor. We also realize the dilution of the enzyme inhibitor complex, and we observed that the restoration of the enzymatic inhibition, indicating that our compound is a reversible inhibitor. So concerning now the naphthol series, we use the same synthetic strategy, as you can see here. We can use some commercial compound, but some naphthol compound were synthesized using this synthetic pathway. So we realized in the first step Friedel-Crafts acylation using benzene derivative and succinic anhydride in presence of Lewis acid to obtain the corresponding acylated compound. The ketone group could be reduced using a Clemmensen reaction to obtain this acid compound, which could be cyclized using polyphosphoric acid. And we obtain the tetralone, which could be then functionalized at position alpha of the ketone group to introduce the ester function. We obtain a mixture of keto and enol compound. This mixture is then treated by copper iodide, to obtain the naphthol compound, and the ester function could be saponified using potassium hydroxide. We also prepare some isomer of position, as you can see here and here. So we also tested this compound against Kallikrein 6 at 10 micromolar. In some cases, we were not able to determine the percentage of inhibition because some compounds here are autofluorescent. So this compound interfere with our enzymatic test. And for the other compound, we observe that 1-hydroxy-2-naphthyl and 3-hydroxy-2-naphthyl are interesting. And we observe the better activity with MIT5 with an IC50 around 1 micromolar. Like before, we modulate the concentration of the substrate and the concentration of the inhibitor to obtain the Dixon representation. And as you can see here, there is no variation of Michaelis constant, so it could be significating that this inhibitor is a non-competitive inhibitor. So the mechanism of binding of this compound is probably more complex than for the other compound in the phenol series. We also observe that this compound is a reversible inhibitor. So to resume, we identify 5 different interesting compounds with an IC50 below 10 micromolar. We have also tested the influence of the hydroxy group by removing it in the structure of MIT5, and we observe the loss of activity. It's not surprising because this hydroxy group is involved in several key interactions with the target. We also change the hydroxy group by NH by synthesizing this endoleic compound, or removing the hydroxy and replacing it by a ketone group. But in the 2 cases, we lost the activity. So the hydroxyl is very important. We also remove the carbonyl group here, and once again, we observe a loss of activity. So to resume, we can introduce alkyl group at position 4 and 5. We can introduce naphthyl group in here or here, and we cannot change the hydroxyl and the carbonyl group. We made some docking, some in silico analysis with these two compounds. So the orientation of these two compounds in the active site of the enzyme is quite similar. And the benzamine group pointing to the S1 pocket with the primary amine, which interact with the aspartic acid residue at the bottom of the S1 pocket. The hydroxyl group here is also involved in polar interactions with some amino acid of the enzyme. And finally, the NH group of the amide function do also an hydrogen bond with the catalytic serine of the enzyme. We also tested these two compounds against other serine proteases, and we obtained some activity against Kallikrein 1 and plasmine. So these two proteases, as I said before, are involved in the proteolytic network. So it could be interesting to have compounds which could be able to inhibit these different proteases. But we need to check if inhibition of this enzyme could not lead to a side effect. These two compounds were also tested on several other proteases, as you can see here, and we didn't see any significant inhibition at 10 micromolar. Perhaps only with MIT5, we obtain a little effect on Kallikrein 8. So we need also to check if it's not problematic. We evaluated the potential cytotoxicity of our compound on two different neuronal cells, cortical neurons and striatal neurons at different concentration. And we observed that the terbutyl derivative is toxic at 25 micromolar in the two different neurons, whereas the MIT8 and MIT5 are toxic at higher concentration, around 100 micromolar. We have also a little effect at 50 micromolar in the two different neurons. So we can conclude that these two inhibitors did not show significant toxic effect and were selected for further studies. So we then evaluated the potential pro-differentiating effect on oligodendrocyte. So multiple sclerosis is characterized by the demyelination of neurons, and in CNS, myelination requires mature oligodendrocyte to create the myelin shields. So oligodendrocyte, mature oligodendrocytes are produced from OPC, so oligodendrocyte precursor cells, which could then be differentiated into mature oligodendrocyte. Our biologist partners have developed a test to measure the pro-differentiating effect. They developed a cell line which could express green fluorescent protein at all stages of the development of the oligodendrocyte, and only the M-cherry at the last stage. So by evaluating the fluorescence, we can measure the quantity of mature oligodendrocyte and the pro-differentiating effect. So we obtain this result. So best results were obtained with MIT8. We can see an increase of twofold of the fluorescence compared to the control. So we can conclude that we have a pro-differentiating effect at this concentration. When we increase the concentration, we observe a loss or reduction of the pro-differentiating effect, probably due to some toxic effect in this cell line. With MIT5, the effects are less marked, but at 5 micromolar, we observe a significant increase compared to the control. This differentiating effect was also evaluated on other model using primary culture of rat OPCs. So quantification of MBP, so myelin basic protein, the main component of the myelin, was done after incubation of our compound, and the results were compared to the untreated OPC. So we can see that these two compounds could induce differentiation with more MBP detected. It is the green spot here. So in conclusion, these two compounds could promote differentiation of OPC into mature oligodendrocytes. The myelinating effect was determined also in cerebellar slice. Slice were fixed after 10 days, so slices are treated by our compound or by the control and fixed after 10 days and stained for MBP, so the myelin basic protein, which here appear in green here, and also stained with for calbidin, which appear in red. Calbidin was used as a marker of axon. So we can see that in the presence of our compounds, we observed an increase of MBP compared to the untreated cell, and the increase was around twofold and equal to cis-retinoic acid, which is used as a positive control. So we can conclude that MIT5 and MIT8 significantly increase myelination. So to finish with this compound, we determine some important parameters. Solubility at different pH value was measured. We have acceptable solubility at acidic pH for both compounds. So, however, MIT5 show poor solubility at neutral and basic pH, which is not the case with MIT8. We observe that MIT5 binds moderately to plasma proteins. We didn't observe some problem with metabolic stability. We have good metabolic stability with human liver microsomes, and also good permeability using the CACO2 test model. Finally, no ERG inhibition have been detected with these two compounds. So now we want to test both compounds in vivo using a multiple sclerosis model of mice. And we work also to increase the inhibition potency and also the selectivity of this compound. So to conclude and to finish, I would like to thank all the people involved in this work. So the chemist, in particular, Dominique Arama, who worked on Kallikrein 7 during his PhD, and also several students who works on the Kallikrein 6 inhibitors. So all the chemistry was done in Montpellier, in the Institute of Biomolecules Max Mousseron. And we recently moved to a brand new building, as you can see here, which is really nice to do some exciting chemistry. So I would like also to thank my colleagues and in particular Professor Shahrazad El Amri and all her team for the determination of the activity of our compound, and also Dr. Brahim Naït Oumesmar and Cyril Debout from the Brain Institute of Paris for all the experiments of differentiation and myelination. And finally, I want to thank all for your kind attention. So thank you.
Thank you so much. Thank you so much, Professor Masurer, for this very interesting talk. Actually, very nice talk.
Thank you.
And this is among the very few options or possible options for treatment of MS. So we know about cortisone, but we don't know about kallikrein inhibitors for treatment of this serious disease. So I hope you success in your next step in vivo and later. Yeah.
Yeah, thank you. That yeah, the problem of this enzyme is probably the selectivity, because there is a lot of isoform and they share some common characteristics. So it's difficult to have some selectivity in this family of enzyme. So we hope that we will have some good results.
Hope so as well. Yeah. So now the floor is open for questions. So, questions? Just a minute. Okay, then I have something to ask about from my side, Professor.
Yeah.
For this 2-hydroxyl group, it looks very important for activity. So if you look for this compound, it's 2-hydroxy with benzamide on the next, with amide on the next carbon.
Yeah.
Is there a chance to have like a rigid or cyclized derivative of these two groups next to each other?
You mean the hydroxyl group could cyclize
And the amide, yes.
and the amide?
Yeah. Because sometimes there is what is known as a pseudo ring. When you have hydroxyl and amide next to each other,
Yeah.
there is a chance for intramolecular hydrogen bonding
Yeah.
to form
Yeah.
what is known as a pseudo ring. So some people in this case will replace this by a cyclic derivative.
Yeah, we didn't try it, but yes, it's a good things to try. When only what we have done is a modification of the hydroxyl or hydroxide group by NH or ketone, as I show you, but we didn't go further, and we need perhaps, yes, to try to introduce another cycle to this part.
Okay. Okay. Other questions? So I can see a lot of thank you and very good words about the talk. So by this, Professor Sameh, do you like to say a final word?
Thank you, Professor Ashraf. Thank you, Nicolas, for the interesting talk. Actually, it was enjoying and very elaborative. And as Professor Ashraf said, it's opening ways for treatment of one of the difficult diseases, which is spread around the world. So we hope we can find really a real solution with this compound or others to stop this.
Okay, thank you.
So,
Professor, yeah, may I ask you, you have published this work in GMET-Chem, and I, of course, understand what is meant by publication in GMET-Chem. But did you patent this compound before publishing or not?
Yes, exactly. Yes, we patent these two
Yeah.
compounds before publish it. Yeah, yeah, yes, exactly.
Yeah, because it deserves to be patented. Yeah.
Yeah, yeah, yes,
Okay.
yes.
Okay. Thank you so much. So now, Professor Samah, do you like to conclude or should I conclude?
You can do it.
Okay. So once again, I would like to thank Professor Nicolas Masurer from Institute of Biomolecules in Montpellier, France. Thank you very much. It has been a pleasure having you with us today. And I hope that this will lead to more collaboration between you and colleagues in Helwan University and the other places in Egypt as well.
So thank you again for this invitation. It was a pleasure to be with you today, this evening, and hope we can see perhaps in the future in Cairo or in Montpellier. Why not?
Yeah, we want it physical, not virtual next time. Okay.
Hopefully we can host you here in Cairo. And at the end, once more, I would like to thank you and thank you, Professor Ashraf, actually for the nice moderation. And I would also like to invite all participants for the next Let's Talk Science. It will be on 14th of March, 8 p.m. as well, under the talk Serious Games, teaching therapeutic decision making in pharmacy curricula by Professor Stephen Duffold from New Zealand, Otago University.
Okay. Good luck, Professor.
Thank you, Dr. Ash.
Okay. Okay. Thank you all so much. Until next time, inshallah.
Inshallah. Thank you. Bye bye.
Thank you. Bye.
Thank you. Bye.
Bye.
Bye.